please repeat that. I'd be happy to do that. Okay? All right. So, in the ileum, which is the terminal part of the small intestine, we have a special structure which is a group of lymph nodules, these masses of B lymphocytes, which we call Peyer's patches. We'll get to that. It'll be on the screen a little later, so you don't have to write it down right now. So that's what you're seeing here. All of this stuff is a lymph nodule. These are B lymphocytes, and notice that it's located in the mucosa of the small intestine. Under the Peyer's patches, we have a special kind of cell which is only associated with lymphoid tissue, and that's called the M cell. M stands for the word microfold. And the reason why they're called microfold cells is because on their lower border, they have these indentations where they sort of engulf. They don't really take the cells in, but they cover some of the immune cells. So they sort of are like a mother chicken taking the little babies under her wing. That's what these M cells do with other types of immune cells, because what the M cell is ultimately going to do, it samples the contents of the digestive tract, and when it detects antigens, it captures those antigens and then brings them in and gives them to the immune cell so the immune cell can respond to them. So in a sense, they're sort of like an antigen-presenting cell, but they have a different morphology than macrophages and other cells that act as antigen-presenting cells. So they're not antigen-presenting cells, but that's sort of their function. They capture antigens from the gut content, and then they take it and give it to the other immune cells that will then produce an immune response. So they're located in the mucosa of the ileum, overlying the Peyer's patches. This is only in the ileum. And they have a pocket formed of invaginated membrane that contains lymphocytes, macrophages, and dendritic cells. And so they capture intestinal microorganisms and antigens, transfer them to the immune cells that they are sheltering. So they're a very cool type of cell. We also have M cells occasionally in other organs like the lungs if you have lymph nodules in the lungs. So wherever you have a lot of lymphoid tissue, you may have M cells associated with it. Okay, let's talk about the lamina propria. It's just typical lamina propria. It forms the core of the villi, carries blood vessels, lymphatics, nerves, and so forth. Now, this is a diagram of a villus. So you see that let me go back and use my mouse again. I'm sorry. I need to get new batteries. I should have done that during the break. So these are our simple columnar epithelium. This is lamina propria in here. And notice that there's a blood vessel network. But there's also in the center a huge lymphatic capillary. And we call this a lacteal. What does the prefix lact mean? You've heard of lactose. What is lactose? Sugar found in milk. So lact means milk. So does that mean there's milk in these things? No, it does not. The reason why they're called lacteals is, again, a historical reason. When people first examined these, there was a milky-looking substance in them. And so they said, oh, this has milk in it. We're going to call these lacteals. It's not milk. It's emulsified fat is what it is. So as the enterocytes are taking up digestive material, the fat gets put into the lymphatic system. The other nutrients, such as the amino acids and the carbohydrates, go into the bloodstream. And so, for whatever reason, the fat goes into the lymph rather than the blood, and then it's carried by the lymphoid system, eventually into the bloodstream later on. So lacteals are special, large lymphoid capillaries that are found in the lamina propria of villi and are involved with absorbing fat from digestive material and then transporting it to wherever it needs to go. Muscularis mucosae is typical, separates the lamina propria from submucosa. So once again, this is a villus. You can see that there's lots of goblet cells. The little white dots are the goblet cells. The other cells are the enterocytes. This is lamina propria here. And this is our muscularis mucosae here. We can see submucosa in the core of this plica and submucosa down here. And so I hope that that's clear to everybody. All right, submucosa is connective tissue that forms the core of the plica, separates the mucosa from the muscularis externa. It has large lymph and blood vessels and so forth. Now, in the duodenum, and only in the duodenum, so this is one of the identifying features of duodenum. You will be tested over this. There probably will be a picture of duodenum on the final exam, and you will know it is duodenum because you will see villi, you will see crypts, you'll see Paneth cells, and you will also see huge mucus glands. And so here are the mucus glands. Notice they're in the submucosa, but they spill over into the mucosa. And notice that this mucus gland in this particular picture has penetrated the muscularis mucosae, has invaded the lamina propria, and it's going to release its contents up onto the surface, into the lumen. So the only part of the small intestine that has these huge mucus glands is the duodenum. Why do we have them there? Because the contents of the stomach that's being spilled into the duodenum is highly acidic. The chyme, remember, has a pH of somewhere around 2, between 2 and 3. And so that acid would damage the small intestine. So these mucus glands produce an alkaline mucus that neutralizes the acid coming from the stomach to protect the small intestine and the rest of the digestive tract from that acid. So we call these Brunner's glands. They're a mucus gland, and they're only found in the duodenum. Okay, Peyer's patches, which we just mentioned, are only found in the ileum. Now, careful, because that doesn't mean that you won't occasionally see a lymph nodule in a different part of the digestive tract. The difference is that in the ileum, it's always there, and there's always massive amounts of it. It's huge. You can see Peyer's patches theoretically, I've never tried this, by opening up a small intestine and just looking, and you can see this bulge where the Peyer's patches are, supposedly. I've never done that, I don't want to, but supposedly that's the case. So Peyer's patches are huge aggregates of lymphoid tissue found in the terminal part of the ileum, just before the ileum empties into the large intestine. And so if you see the Peyer's patches, you will know that you're in ileum. If you see the Brunner's glands, you will know that you're in duodenum. If you know that it's small intestine, but you don't see Peyer's patches or Brunner's glands, then just call it small intestine. Okay? You don't have to specify which part on the practical. All right, the muscularis externa has an inner circular layer, an outer longitudinal layer separated by myenteric plexus, and the small intestine, for the most part, is covered by serosa. All right, and so here is a nice picture of the wall of the small intestine. Another John Huff picture. John, you're amazing. Thank you. These are much better than any of the pictures from the textbooks. So this right here is a plica. What is in the core of the plica? What's that? Submucosa. Submucosa. Okay. Now it's kind of hard to see, but this is the muscularis mucosae right there. All of this stuff right here, the little granular looking stuff, that's all lamina propria. And down here, we have the crypts with the Paneth cells, and here are the villi sticking out from the surfaces. All right, and then here's submucosa here. Here is the inner circular layer of muscle. This is the myenteric plexus right there, so it's easy to pick out. And then this is the outer circular layer, and then out here is the serosa. So this is a great picture because we see all the layers in this picture. Now it's low magnification, obviously, but you can pick up all the layers in this picture. So it's an amazing picture. Okay, let's go to the large intestine, also called the colon. It's formed of 6 regions. I think Dr. Escobar already went over these with you in her lecture. So remember, there's the cecum that has the ileocecal valve, and that's the connection between the small intestine and large intestine. We have the appendix hanging off of the cecum. We looked for that the other day. Some cadavers had it, some didn't. And then we have the ascending colon. We have the transverse colon coming across. We have the descending colon coming down. Sigmoid colon. Sigmoid means shaped like the Greek letter sigma. A capital sigma looks like an S. And so it's named for its S-shaped curve. And then that leads into the rectum. Rectum means straight. Rectitude is being straight, being right, being just. Okay, so rect. And then that leads to the anal canal. All right, so the primary roles of the colon: to reabsorb water and electrolytes and to form fecal material, feces, from the indigestible material. So this is what the wall of the small and the large intestine looks like. This is the diagram from your textbook, and here's an actual photograph. Notice we still see the crypts, just like we saw in the small intestine, but there are no villi. No villi, only crypts. And most of the epithelial cells are goblet cells. Now there's enterocytes, they call it colonocytes or something. There are other cells, but goblet cells tend to be the predominant cell type, and there are no Paneth cells. Okay? All right, so the mucosa, epithelium, is simple columnar, made up of absorptive cells called colonocytes, and predominance of goblet cells. No villi, just crypts. Remember that. Cells are interspersed with neuroendocrine cells and stem cells. The appendix is an extension of the cecum, and it has lots and lots of lymph nodules, but no absorptive function. Now, in young people, the appendix is highly lymphoid. So if you look at the appendix of a child, you'll see tons of lymph nodules. It'll look very much like Peyer's patches. How will we know that it's not Peyer's patches? Because we don't have villi. We don't have Paneth cells. We just have crypts. So the histology of the appendix is like the histology of the colon, except for oodles and oodles and oodles of lymphoid nodules. All right, lots of masses of B cells. As we get older, the lymphoid tissue sort of disappears. So if you looked at an old person's intestine, there wouldn't be that much lymphoid tissue anymore. Old people don't have as strong of an immune system as young people do. So that's why appendicitis is typically a childhood disease. Usually it happens in kids. We had a professor in the department, he was a neuroscience professor, and he got appendicitis at the age of 82, which is really unusual. So we said that he'd entered his second childhood. He had a childhood disease. Okay, the lamina propria is between the crypts, typical, but it has lots of lymphoid tissue gesundheit. So MALT just means lymphoid tissue, mucosa-associated lymphoid tissue. And the muscular mucosa is typical, and the submucosa is typical, and the external, now there's an intercircular layer which is normal, but as Dr. Escobar pointed out, the outer layer is not continuous, but there are 3 bands of longitudinal fibers. And what do we call those bands of muscle in the colon? She pointed them out in her lab demo the other day, I know, and I think some of us look for them in lab. So what are those bands of longitudinal fibers in the colon called? Remember? It's the same as the scientific name for tapeworm. Does anybody know what the scientific name for tapeworm is? Tinea. Tinea means tape. Okay? So these bands are called tinea coli. All right, so the outer longitudinal layer is confined to 3 longitudinal bands called tinea coli. You can see them here. And here's a nice example right there. So they're evenly spaced around the colon, and they all converge at the appendix. So the other day I was on one of the cadavers with you guys, I don't remember who it was. We were looking for the appendix, and so I said, well, let's find the tinea and trace those down, and where they meet is where the appendix is supposed to be. That person didn't have an appendix because they'd had an appendectomy, so we couldn't find the appendix. All right, the anal canal is the distal end of the GI tract. Simple columnar epithelium of the rectum transitions to stratified squamous because the rectum, the anus is going to continue into the skin. So first you have non-keratinized stratified squamous, and then transitions to keratinized as you go to the skin. And that's all we're going to say. We're not going to say any more. There are some sphincters there. Here's some pictures. And thank you, Lord, for the sphincters. Okay. And that's the end of this. Oh, I am an art lover, and I was just in a wonderful museum where I saw these beautiful glass sculptures. Anybody know where this is? Obviously nobody's been here. This is Seattle. Anybody from Seattle? This is right next to the Space Needle. There's the Chihuly Museum, Glass Museum. Have you been to it?
Yeah, sometimes they hold yoga in there.
Yeah, it's spectacular. If you ever get to Seattle, go to this museum. It's just gorgeous. Okay, so that's what it looks like. Okay, we're going to take a micro break while I switch lectures.